So good afternoon, everyone. I'm Sabina Halappanavar, a research scientist from the Environmental Health Science and Research Bureau of Health Canada at Ottawa. I have been investigating the cellular mechanisms of lung fibrosis in the context of exposure to nanomaterials in the lab for many years. So in today's presentation, I will talk about how one can use adverse outcome pathway framework to guide the development of in vitro alternatives to testing fibrogenic substances. So pulmonary fibrosis is one of the adverse outcomes of exposure to environmental toxicants or a side effect of drug-induced toxicity. Idiopathic lung fibrosis is a progressive and fatal disease in humans, the cause of which is unknown. Much of what we know about IPF comes from toxicology studies conducted on the substances listed on this slide, for example, bleomycin, silica, asbestos, etc. So regardless, fibrosis can be defined as overgrowth, hardening, or scarring of tissue, a result of uncontrolled or excessive collagen deposition. Or it can be defined as the final pathologic outcome of excessive collagen deposition. So in general, inhalation exposure to any substance in the environment will activate immune response. And depending on the material characteristics, duration of exposure, or amount of exposure, it will result in tissue injury or varying degrees of tissue injury, leading to activation or of healing response. So this immune and healing response involves a series of physiological and molecular and cellular events, some of which will start within the minutes after exposure, and some manifesting after hours or days of exposure. And at each of these events or during these events, several molecules, including inflammatory mediators, growth factors, or others, are released. Several cell types are stimulated or activated, and dynamic microenvironment is established. So all of these events are causally linked, depicted by the arrows here, meaning each event is required for the other to occur. So the ultimate goal of this cascade is to repair the injury and reestablish the lost homeostasis. However, following exposure to a substance exhibiting material properties that can cause extensive tissue injury, such as in the case of carbon nanotubes, or repeated exposure to drugs such as bleomycin, or persistent exposure, such as in the case of inorganic particles that cannot be cleared efficiently, the acute immune response is not resolved, leading to another cascade of events depicted on this slide, for example, and that cascade involves chronic immune response, activation of fibroproliferation, secretion of fibrogenic mediators, excessive collagen deposition, and eventually leading to fibrosis. So here on this slide is a simple schematic showing cell types involved and the events involved in normal healing process, returning to functional tissue homeostasis, or fibrotic response leading to non-functional tissue. So as mentioned earlier, there are many different kinds of profibrotic substances. And for some of them, you can see the initiating events are described. Regardless of the type of the substance or the initiating event, persistent tissue injury and chronic immune response triggered by the persistent or prolonged exposure seem to be common following exposure to any fibrogenic materials. So as in case of any other disease research, lung fibrosis research also involves human clinical studies. A lot of work has been done in human subjects. Experimental animal models are involved, In vitro studies involving cells derived from human or animal tissues are considered, as well as in silico capabilities exist. However, as shown in this figure, human or the regulatory relevance of the study or the study results increases with complexity of the model used. So a clinical study will have a direct relevance to the regulatory context. However, as shown in the figure, less complex models allow investigation of the underlying mechanisms, and knowledge that is derived from such models will eventually form the basis for alternative testing strategies, enhancing our ability to screen large number of chemicals for their fibrogenic potential in a time and cost-effective manner. So moving forward, even though we have extensive human work conducted, and although we have animal models available, moving forward, more and more focus will be placed on the development and validation of unconventional or alternative toxicity testing methods that are not animal reliant, but are reliable, reproducible, and high throughput. So considering the fact that we have been working with alternative models for years now, the task of validating an alternative test method seems very easy and straightforward, but in reality, it is not. With an ultimate objective of using the data derived from an alternative test method to establish human exposure guidelines or to predict human safety levels, an established alternative method has to be phenotypically anchored against an adverse outcome of regulatory relevance. So on this slide, you will see a kind of roadmap of how one can use alternative toxicity testing method to identify a hazard or assess the risk. And the slide shows the details of a process that is involved. So as I mentioned, before even you start validating a model or identifying an in vitro assay, we have to know an adverse outcome for which an in vitro model is required, or for which we are trying to establish an alternative method, and why. And besides that, we also have to know the endpoint that a particular model is measuring in the context of the adverse outcome that we have identified. Mechanism, the detailed mechanism has to be characterized. The model has to be tested for its performance, meaning its reliability, relevance, and reproducibility, etc., have to be established. And once you have all of that, you can use the validated model and the targeted in vitro assay that has a test guideline now available to derive a point of departure for a particular chemical. And then in consideration of other sources of information, you can move on to deriving the human safety levels. So in essence, the task is really tedious, and a prerequisite may be that we have to establish a mechanism of action. In other words, an adverse outcome pathway with an adverse outcome of regulatory relevance identified and the various key events defined and identified in the path. And this is the key for the successful design of an alternative testing method or a strategy. So then, what is an adverse outcome pathway? Really, in brief, adverse outcome pathways are conceptual. They portray existing knowledge. They represent series of measurable, observable, and essential biological events that occur in sequence following exposure, linking an early initiating biological event to an eventual adverse outcome. The key events share quantitative relationships and are biologically plausible and empirically supported. And the key event relationships or the quantitative relationships are defined by the arrows between the two events. They are not really chemical specific, rather they are driven by the adverse outcome itself. And there can be many adverse outcome pathways, but every single AOP is a linear module of events leading to an adverse outcome. And one can develop many AOPs, and the complexity or the complex biology of an adverse outcome can be represented by the network of AOPs, where more than one AOPs are connected by the key events involved. So AOPs can be qualitative, they can be semi-quantitative, or completely quantitative. And depending on whether or not they are quantitative, their use in the regulatory context differs. So on this slide, you can see the various ways in which adverse outcome pathways can help inform regulatory decision-making. For example, qualitative or semi-quantitative AOPs can inform mechanistic information. They can help discover new biomarkers. They can support improvisation of the existing in vivo or in vitro test guidelines. They can help establish new test guidelines, in vitro or in vivo. They can improve the parameters used in the QSAR modeling. The data can be used for prioritization purposes or categorization of chemicals or substances. They can be used to support read across or weight of evidence in the regulatory decision making. However, if the AOP is quantitative and in consideration of other data sources such as on dose response, physical characteristics of substances, as well as exposure, one can use the information then directly in predictive modeling as well as in support, and the data generated can be used for the risk assessment of the substances. So coming back to lung fibrosis as a disease of regulatory and human relevance. As mentioned in my introductory remarks, I have been investigating the molecular mechanisms of lung fibrosis in the context of nanomaterial exposure. Lung fibrosis is one of the consistently observed adverse outcomes in animal models following exposure to certain types of nanomaterials. Considering the sheer number of nanomaterials that are out there that require immediate assessment and their diversity, and considering the fact that lung fibrosis is one of the commonly observed outcomes, our goal was to develop an alternative testing strategy for lung fibrosis. However, as I mentioned earlier in the previous slides, the road is really tedious and difficult. And before we get into coming up with a strategy, an alternative strategy, we have to be sure of an adverse outcome pathway for fibrosis. So to that end, my group at Health Canada, in collaboration with researchers at PETA and ICAPO, ventured into establishing an adverse outcome pathway for lung fibrosis, an initial and critical step in the development of alternative testing methods or strategy for regulatory purposes. Using the data generated in-house, at Health Canada, and the reports published in the literature, we identified several key events that are commonly induced by several fibrogenic substances. And these include, as shown on this slide, chronic tissue inflammation, persistent tissue injury, activation of adaptive immune response, loss of tissue barrier, and inability to restore the homeostasis, and activation and proliferation of fibroblasts or fibroproliferation, and excessive deposition of extracellular matrix. And all along, it was very clear for us that the KEs, or the key events, are temporal in nature. They involve multiple cell types. They are complex, and there's a complex and dynamic microenvironment involved in all of this. So with that knowledge, the exercise resulted in an adverse outcome pathway that is presently under review by the OECD AOP committee. So this AOP, in brief, suggests that the interaction between the fibrogenic substance and cellular membrane leading to perturbed homeostasis provides a trigger to initiate the disease cascade, which then involves acute other key events, including acute inflammatory and fibrogenic immune response, irreversible tissue injury or excessive tissue injury, chronic or adaptive immune response, and fibroproliferation, where you have fibroblast proliferation and myofibroblast phenotype, and then eventually excessive deposition of collagen. While this AOP is applicable to many substances, it is particularly relevant to those that are insoluble, biopersistent, with or materials with special physical chemical properties that trigger an immune response acutely after the exposure. As you can see, and as it's defined here, the various key events span the different phases of the disease and involve, by the nature, different cell types. And so it is really important to note that a single key event may not be possibly sufficient to predict the disease on its own. And at the bottom, it's shown that quantitativeness of any of these MIEs or KEs will determine its predictive capacity or its use in direct risk assessment of fibrogenic substances. And regardless, what's important to note is that combination of key events may be necessary and have to be considered in a defined or a tiered approach or a tiered testing strategy to eventually predict the lung disease. So having an AOP established for lung fibrosis, which may be tentative at this point and may not have the quantitativeness that is required for the regulatory application, we have served the literature to list the available in vitro assays or methods that can measure the key events identified in this AOP. So you can see the key events, and at the bottom, you can see in the boxes various in vitro assays that are presently available or used to measure several of these key events, or specific key events, I must say. Again, one single assay will not do the job. We will have to consider multiple assays spanning more than one KE to form a basis for a defined or a tiered approach. And in all of this, selection of the cell types or the exposure model is very critical. And just for an example, one defined approach could include epithelial cells or combo of epithelial and macrophage cells to measure the first few KEs, which includes the MIE, that's in the green box here. And then you have immune response key events that are immediately after the MIE. And you can include fibroblasts for the later KE, which will involve fibroblast and myofibroblast proliferation assays. And the various assays in the combo could include some from the initial KEs and the ones later on. And in this specific defined approach example, we are suggesting consideration of the assays that are in red in each of these boxes. So again, tiered or combinations or defined approaches is a key. And more than anything, the selection of right type of cell types and the exposure model will be critical. And these are the aspects, I think, that are addressed in the other webinars in this particular series. So in conclusion, we have been using in vitro systems and models for decades in toxicology in general. However, the relevance of the results derived from such models to in vivo phenotype is not really established, or for most part remains unknown. largely because the past efforts have focused on replacing one in vivo test method with one in vitro method, and that needs to change. And we have to be aware that it requires multiple assays. So, in order to have a successful alternative testing strategy, the strategy needs to be anchored to an adverse outcome pathway or to a mechanism that is well established and empirically supported. And we have to be able to define what we are measuring with the alternative test method in the context of the adverse outcome that we are trying to predict. Since complex diseases involve complex biology, more than one type of in vitro assays measuring more than one key event spanning the different phases of an adverse outcome pathway, as shown in the previous slide, will be needed. And the extent of quantitativeness of key events and thus the AOP will determine its applicability to regulatory decision making. So we have, in this presentation, we have provided AOP 173 as an example. However, please note that it is a tentative AOP and quantitativeness of this AOP is yet to be established. And when that quantitativeness is established, it can be validated and adapted for further in test guideline development and for predicting or replacing the animal test strategy. With that, I would like to thank my host organization, Health Canada, for sustained financial support, and my co-authors of the AOP 173 for their contribution. Thank you.